Hi guys, let's talk about the Calvin cycle. So Calvin cycle can be broken into three stages: carbon fixation, reduction, and regeneration. And they are color coded there for you to see on that diagram. Carbon fixation stage is just what it sounds. We're going to take a carbon dioxide and we're going to fix that inorganic carbon into an organic molecule called RUBP. The RUBP is five carbons, which is shown by the five purple circles there. The CO2 is one carbon. When you add it on, you make a six carbon intermediate that's very unstable. But then that will break up into two three carbon molecules, which we call glycerate 3 phosphate, which is going to be the ultimate product of the carbon fixation stage. Two glycerate 3 phosphates per RUBP and per CO2. The enzyme that's responsible for this is one of the most famous enzymes and the most ubiquitous enzyme on the planet called Rubisco. Rubisco stands for RUBP carboxylase oxygenase because you're going to either be adding carbon dioxide onto an RUBP, which is the carboxylase function, or we're going to see later on it can actually add oxygen on as well, unfortunately, which is the oxygenase function. Now, if you look at the first molecule on the left, that's your RUBP. There's five carbons. You can't see the carbons because they're shown as the points of the molecule, but everywhere where there's a little bend in that molecule, which you'll see there are five bends on there, are going to be five carbons. When you add in that carbon dioxide, you make that unstable intermediate, which then breaks apart on its own into two molecules of glycerate 3 phosphate. Glycerate because it's a carboxylic acid group at the end, that's C double bonded O to another O minus that has lost its proton. And so we have two of those glycerate molecules. That is the first step. We fixed our carbon. Now, RUBP, like we said, is a really important enzyme, right? And as enzymes go, it's not very specific and it goes painfully slow. Usually, an enzyme can make thousands of molecules per second, but RUBP can only make about three glycerate 3 phosphates, or can only fix about three carbon dioxides per second. That's very slow as enzymes go. And because of that, basically the chloroplast is just filled with a ton of RUBP, not RUBP, a ton of Rubisco, which frankly, the most abundant enzyme on the planet is Rubisco. Now, Rubisco actually evolved before we call the Great Oxygenation Event. The Great Oxygenation Event is when plant life, early plant life, what led to the plants that we see today, made all the oxygen that's in our atmosphere. The early atmosphere had a lot more CO2 in it than it does today, and a lot less oxygen. So when we talk later about some of the shortcomings of Rubisco, it's important to know that it evolved at a time well before what we have now. And it's been good enough for the plant system to survive, right? So remember, evolution does not make things perfect. All you have to do is get an adaptation that allows you to survive in a point in time, and then if it's good enough later on, you can carry it along. But Rubisco itself has some shortfalls, which we're going to see later on when there's too much oxygen, which is a process we call photorespiration. But that's a little bit later. Now, the second stage of the Calvin cycle, right, if I go back to right here, this red stage, so highlight it, it's called reduction. And in the reduction stage, what we're going to do is we're going to take that glycerate 3 phosphate, which you can see that last carbon is bonded to an oxygen, actually double bonded to an oxygen and then bonded to another oxide. And then what we're going to do is we're going to add a phosphate on there so that we can destabilize that bond, right? Make it more likely to react. And then we're going to replace that with a hydrogen. And remember, whenever we're increasing the carbon to hydrogen bonds and decreasing the carbon to oxygen bonds, what we're doing is reduction. So NADPH is going to provide the electrons and the hydrogen needed to reduce the glycerate into a glyceraldehyde. That is now an aldehyde because it's going to be a carbon double bonded to an oxygen, which is then bonded to a hydrogen, not an 8, right? Not a glycer 8 because it no longer has that second oxygen. Since we've now increased the carbon hydrogen bonds and decreased the carbon oxygen bond, we have reduced the molecule. The electrons and the hydrogens came from the NADPH. The ATP that drives the reaction was also produced in the light dependent phase, just like the NADPH was. Again, this is just showing a detailed view of the two differences of the molecules, the phosphoglycerate, which is also another way of saying 3-phosphoglycerate or glycerate 3-phosphate. They're all the same thing, just rearranging of the words parts. But that's the top molecule. That is the not reduced version, that's the oxidized version. And then the one below it, the glyceraldehyde 3-phosphate, that now replaces that carbon-oxygen bond, which you see in the first molecule, with a carbon-hydrogen bond. That's what we're doing in this reduction stage. Now, at that point, once you make your glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate is also just called triose phosphate in the IB world and elsewhere, but really in the IB world. That glyceraldehyde 3-phosphate is the product, right? We're done. But I need you to know that that's not glucose. It actually takes 2 turns of the Calvin cycle to make a glucose because each one only makes a triose phosphate. But also that glucose is not the only thing that can happen. Basically, if you see this diagram to the right, we have CO2 coming in to make your glycerate 3-phosphate. That's before we've reduced it. And then that particular molecule can go down different pathways. We can turn it into a pyruvate and use it in the mitochondria to do cellular respiration stuff. We can then let it go become triose phosphate, which can then become a glycerol potentially if we want to put it down that pathway. We can combine triose phosphate to become sugar molecules, 6-carbon sugars, which can be like a glucose or a fructose. We can just make glucose from it. We can make glucose and fructose come together to make sucrose, or we can make large polymers of glucose like starch and cellulose and so on and so forth. Right. The interconnectedness of these metabolic pathways are a really important concept now that you're in HL biology. Right? If we want to make proteins, well, proteins need nitrates from the soil, right? Because we're going to need to make ammonia for our the amino acids, but we're also going to need the carboxylic acid side of those proteins, right? That carboxylic acids, that's like a pyruvate, that 8 part, that acetate. Those are the carboxylic acid sides of those compounds. So all these metabolic pathways are interconnected, as we've sort of seen throughout this year. Now, once you've made this triose phosphate, you're basically done with the intended product, but this is a metabolic pathway that's cyclic. So we need to regenerate our RUBP so the process can continue. And while this may look pretty complicated at first, you have to remember that when we do 2 turns of the Calvin cycle, we basically take 6 carbon dioxides, and from that, we're going to get 6 triose phosphates ultimately, right? But only one of those 6 is going to be we're going to take 3 carbon dioxides per cycle. Per cycle is 3 carbon dioxides, and you're going to end up with 6 triose phosphates. Because when you have the 3, if you look at this diagram to the left, you see those green circles. You have 3 5-carbon RUBPs. We have 3 carbon dioxide. They come together and make 6 glycerate phosphates. Glycerate or 3-phosphate or glycerate phosphate, they basically just different ways of saying the same molecule. And then what happens is when that gets reduced, we're going to get 6 of our glyceraldehyde phosphates or glyceraldehyde 3-phosphates. Of those 6, one of them is the product. The remaining 5 go off to regenerate the RUBP so the process will continue. So how does this happen? Well, it happens in a series of reactions. If you look down here, the C3 that is highlighted in red, that is going to be your glyceraldehyde 3-phosphates, right? One of the 6 is your product, but the remaining 5 have to regenerate it. So how does that happen? Well, you get one and another one. You write a C3 and a C3 come together. It makes a 6-carbon molecule that we're not going to name. That 6-carbon molecule can then go on to the next reaction, where it can combine with another one of those glyceraldehyde 3-phosphates, the other C3, to make a 9-carbon molecule. Again, we're not going to name that molecule. That 9-carbon molecule then spontaneously breaks apart. It's too big, it's unstable. And it's going to break apart into a 5-carbon molecule, which is actually going to be our RUBP, and then a 4-carbon molecule that's going to go on to reaction 4. That 4-carbon molecule is going to combine with another one of our glyceraldehyde 3-phosphates, another one of our 3-carbon triose phosphates molecules, right? One of those products. And that's going to make a 7-carbon molecule. That 7-carbon molecule then combines with another one to make a 10-carbon molecule. That 10-carbon molecule then breaks apart into 2 more RUBPs. So at the process, throughout those 6 reactions, we've now generated 3 RUBPs, right, which are seen by the C5s that are highlighted in green, and we've used up 5 of our triose phosphates, which are the C3s highlighted in red. Where if you look there, 5 molecules that are 3 carbons 15 total carbons, is the same amount of matter as three molecules that are 5 carbons, also 15 carbons. They're just rearranged into now the REBP form, and now the process of the Calvin cycle can continue. Again, we talked about Rubisco is not a great enzyme because it's so old, it lacks specificity, and it evolved before there's oxygen. So RUBP can have two fates when it's combined with carbon dioxide, which is what the plant wants, or oxygen, which is what the plant doesn't want. Rubisco doesn't really tell the difference between them because they're both small, non-polar linear molecules. There's not a very big difference between their structure and their chemistry, even though it matters greatly which one you add. So basically, when you have low temperatures or a high carbon dioxide to oxygen ratio, that's a good thing. We do the Calvin cycle. When temperatures are too high, or you have a low carbon dioxide to oxygen ratio, we can do this thing called photorespiration. Photorespiration is going to ultimately oxidize RUBP, kind of the opposite of what we want to do. We want to ultimately go ahead and take that RUBP, right, and add carbon dioxide to it and then reduce that into sugar so we have stored potential energy. This does the opposite. It's going to actually cause RUBP to break up into a 3-carbon molecule, which you want, and then a 2-carbon molecule, and basically slowly oxidize that RUBP, which is not good. Now, if you look, RUBP has 5 carbons, which you can see there. In normal carbon fixation with CO2, because you're adding on another carbon, you get 2 3-carbon molecules, which are both the 3-phosphoglycerates, or glycerate 3-phosphate, same thing. That's your GP, that's your first product of carbon fixation. That's what we want. We have 3-carbon molecules, 3-carbon molecules, they'll go on, they'll both become triose phosphates. Some of them will be regenerated, some will become sugar. That's all the good stuff. If we have oxygen instead, because we only have 5 carbons, we're not going to ultimately get another 3-carbon molecule. One of them will become a 3-carbon molecule, which you can see at the top, but the other one's only going to be a 2-carbon molecule, right? So ultimately what we're going to do is we're going to lose some carbon-hydrogen bonds if you look up there. Both of those have the carboxylate, right, on the top, right? But the bottom molecule, which is the 2-phosphoglycolate, which you don't need to know that name, it's missing that second carbon. That second carbon that would be carbon bonded to a hydrogen, bonded to a hydroxide. And because of that, that molecule cannot undergo the reduction stage. And so what's going to happen is that molecule is ultimately going to have to be shuttled out. And this is a little bit of a nice to know this part. This is not an IB curriculum, but that phosphoglycolate is going to have to then be shuttled to the mitochondria, where it's going to be broken down and regenerated to serine, and that serine is going to be transferred into a glycerate, and that glycerate is going to then come back, and then now that's going to regenerate some glycerate in the chloroplast. But that whole process is very energy intensive, and so ultimately this is a net loss of energy for the cell, because you're never going to actually be adding on that carbon. You're not going to be reducing it. It's not going to be a great process. You're basically going to do respiration, where you're going to be oxidizing your sugar molecules and your carbon molecules rather than reducing them, which is the whole purpose of photosynthesis. So basically, photorespiration, just a nice little aside, it limits how much plants can produce our sugars, right? How much primary production happens with sugars of our plants. So it's estimated that it reduces the wheat production by about 20%, soybean production by about 36%, merely due to the fact that these plants, this enzyme evolved in a time before oxygen, with high levels of CO2, so it's not going to be great. Now, temperature affects this, right? As you increase temperature, you increase Rubisco's affinity for oxygen, which is a problem, because as we have rising temperatures of our environment, that's just going to cause basically our food production yield to go down, because it's going to cause this enzyme to have more of the photorespiration process that we don't want and less of the photosynthesis process that we do want. Luckily, there are two adaptations that sort of help plants get around this that have evolved over time. They're called a C4 and a CAM pathway. The typical pathway that we see is called a C3 pathway, and it's a C3 pathway because we make a three-carbon molecule when we do carbon fixation. In fact, we make two of them, and that is going to be your glycerate 3-phosphate. But so three carbon C3, that's the normal photosynthesis pathway, the normal Calvin cycle. But some alternate pathways exist. The first one is one where called C4, where a plant will actually segregate where it's fixing its carbon dioxide regularly. Now, what happens in this pathway, you're going to have a collection of cells called the bundle sheath cells. Now, what happens is in the bundle sheath cells, which are really far and really close to the vein, if you look at the diagram of the C4 plant, you can see the bundle sheaths all the way in. They are separate from the mesophyll cells. The mesophyll cells are the ones that are actually going to be fixing the carbon initially that are closer to the opening stomata. Now, what's going to happen is instead of Rubisco doing the initial fixation of carbon dioxide, what happens is there's an enzyme called PEP carboxylase, and that will take a three-carbon molecule called PEP, add a carbon dioxide onto it, and that's going to make a four-carbon molecule called oxaloacetate, which you may have heard of from the Krebs cycle, right? Oxaloacetate. Well, that molecule is then going to be transported to the bundle sheath cells, and then once you have a four-carbon molecule in the bundle sheath cells, one of those carbons is going to come off as CO2. And when that comes off as CO2, that's going to enter the Calvin cycle. And basically what this allows us to do is to manipulate the concentration of CO2. If we can take a whole bunch of CO2 and make these four-carbon molecules where CO2 concentrations might be low, but since PEP carboxylase doesn't have any affinity for the oxygen, or very low affinity for the oxygen, much lower than Rubisco, we can get a lot of that carbon fixed. And then that all goes to the bundle sheath cells, and we release a whole bunch of carbon dioxide at once, more than you would normally get from the atmosphere. And that keeps carbon dioxide concentrations high, which therefore Rubisco doesn't then have the issue of having too high of O2 relative to the CO2, and then we limit the photorespiration. The other option, it's a very similar pathway. This is called CAM pathway. And what this does is pretty much a similar process, but instead of doing it in two different compartments, like the bundle sheath cells and the bundle sheath cells and the mesophyll cells, right? Instead of having two different compartments like the C4 plant, what we get is these plants segregate by night. So at night, when the temperature is really low, because relative to the daytime, nighttime temperatures are lower. And remember, the temperature also changes the affinity for Rubisco. What we're going to do is we're going to have the plants open their stomata at night, allow CO2 to come in during the night, and it's going to fix it into oxaloacetate. PEP carboxylase can work during the night. Rubisco and the Calvin cycle does not work in the dark, right? They need the products of the light-dependent reaction to function. So at night, we're just going to collect as much CO2 as we can while the stomata are open, while it's cooler outside. This also limits the loss of water due to evaporation. So we're going to see this a lot in our succulents and our desert plants. And so what's going to happen is all through the night, the CO2 comes in and we get this PEP carboxylase, the same enzyme as the C4, making a four-carbon molecule, oxaloacetate, which then makes malate. And then that molecule during the daytime, when they shut down the stomata, that molecule releases a flood of CO2 during the daytime. So there's tons of CO2 for doing photosynthesis, thereby also limiting photorespiration. Okay, guys, that's it. It's a lot of information. I'd check out your textbook. Make sure you review these notes. If you need to rewatch this video or ask me questions, feel free to send me a message. Okay, bye.